So here we're going to tap into the potential of adult stem cells. We'll talk about their biology, and we'll talk about these cells as potential for regenerative medicine. So what are adult stem cells? Let's just refresh our knowledge again. These are cells, remember, of the body that can repair tissues when it's injured or when a tissue undergoes normal wear or tear. So some tissues naturally have more stem cells than others and replace its cells continuously. Examples of that would be skin stem cells that regenerate the epidermis and the hair follicles, blood stem cells that are able to regenerate red blood cells and immune cells. And in fact, every 2 weeks' time, you've got a brand new surface of your epidermis, and of course, if you're lucky, your hair follicles not only undergo periods of rest, but they also undergo periods of growth. And blood cells have an enormous capacity to continually regenerate. So other tissues, though, have very few stem cells. Those are the tissues that we were talking about earlier in the presentation, cells of the central and peripheral nervous system. In fact, it's only the last couple of years that we've even known that there are, in fact, stem cells in the adult nervous system. Pancreatic tissue is even more problematic. Doug Melton's laboratory has been studying pancreatic islet formation and found that the only way to get islets is by damaging existing islets and getting islets to produce more islets. That there appear not to be any of these multipotent adult stem cells, for instance, that could regenerate an islet if necessary. And so that's one of the reasons why I spent the first part of my talk discussing embryonic stem cells. But for those tissues that do have an abundance of adult stem cells, we can think about those types of stem cells for regenerative medicine. So the characteristics of adult stem cells then are the ability to divide, to produce more stem cells. So whether they're adult or whether they're embryonic stem cells, all stem cells have self-renewal capacity. And the ability to produce differentiated tissue. In this case, for my laboratory, epidermis or hair or sebaceous glands. One of the characteristics of adult stem cells is they tend to be immature relative to their progeny. So when we study the hair stem cells, those cells are less mature than the rest of the cells within the hair. Adult stem cells are typically multipotent, and they can make a subset of cell lineages, as I've just described to you, in the case of hematopoietic stem cells or skin stem cells. So the cytoplasm determines the gene expression pattern that the nucleus will have. We talked about the fact that the adult skin stem cell has a nucleus and properties of gene expression that are quite different from when that adult skin stem cell nucleus is placed into an environment of the oocyte. In the case of taking the adult skin stem cell, putting it into the oocyte to make a hybrid cell that then goes on to generate embryonic stem cells, the nucleus gets reprogrammed. In the case of the adult skin stem cell, the cytoplasm of this cell is different and presents a different program of gene expression for this nucleus versus this nucleus. So even though this nucleus and this nucleus have the same genetic content, the pattern of gene expression is different. At present, the only cell that we know about and the only cytoplasm that we know about that is capable of producing an unlimited different variety of gene expression patterns afforded to all of the cells of the body is that of the egg cytoplasm. As far as we know, adult skin cytoplasm does not have that capacity. So we'd really like to know more about what's the difference between this cell and why these cells have different patterns of gene expression and just what those different patterns of gene expression are. So my laboratory does study skin, and we use that as a model system. And I think of all of the various different tissues and organs of the body, there's no question that nature clearly has had a lot more fun and fancy in creating body surfaces than she has in creating any of those ugly organs that are tucked underneath it and that most scientists work on. So my laboratory has been fascinated for my lifetime in skin, as is all of nature, and I think many of you. It's the one tissue of our body that we really feel intimate about. We see it every day. We notice when there's even the slightest change in our skin. And then, of course, when you consider that virtually every society of the world has adorned upon what nature has given us. And I think it's kind of interesting that often when you consider different societies and different cultures and different adornments that societies have given to their skin and their hair, that one often uses appendages from other animals, the feather, in many examples, as a further case of enhancement of our skin surface. So that's just one of the reasons why I always like to remind my audience that you too have been interested in skin, and we only go just a little bit deeper into the skin. So where do adult skin stem cells come from? During embryonic development, cells become increasingly more restricted in what tissues they can make. So back in early development, the skin surface begins to form after gastrulation of the embryo. And at that stage, there's a single layer of multipotent embryonic skin stem cells. And these cells can give rise to the epidermis, the hair follicle, the sebaceous gland. In the case of the epidermis, for the unipotent stem cell, those cells exist within the epidermis, and when we go from the single-layered epithelium to the stratified epithelium, this is the structure of the epidermis that is going to be maintained in your adult skin. And the single layer located in the innermost, called the basal layer of the epidermis, is going to be the only layer of cells that remains unipotent and remains proliferative. These cells are going to differentiate, and as they differentiate, they start to move upward, and these cells way at the top are going to be the cells then that ultimately are going to be sloughed from the surface of your skin, continually being replaced by inner cells differentiating and moving upward. So this is the reservoir down here of where the unipotent cells of the adult skin epidermis are going to be. So these are unipotent adult epidermal progenitor cells. These are multipotent embryonic skin stem cells. If adult stem cells reside within a tissue, they're usually tucked as safely and deeply as possible within the tissue. And that's a property of many different types of adult tissues. So stem cells of the skin, making epidermis and hair and more. The skin stem cells provide the nearly endless supply of cells to replenish the epidermis and the hairs of our body. So can we isolate them? Can we coax them to become hair as well as epidermis? Can we coax them to become other types of cells without deriving embryonic stem cells by nuclear transfer into oocytes? So back when I was a postdoctoral student, I began to study skin stem cells, and at that time I was studying almost exclusively human epidermal stem cells. And one of the reasons why I became a skin biologist is because skin epidermal stem cells are amongst the few stem cells of our body that have the capacity to be cultured in the laboratory and to be maintained and propagated as adult stem cells of the skin. Now, I just talked about in the previous part of the presentation embryonic stem cells that we could culture and which maintain their properties endlessly. Adult human epidermal stem cells aren't like embryonic stem cells in that we can't expose them to neural factors and get neurons, but these stem cells can grow and be maintained in the laboratory, and this is how we do it. We take these cells and we place them into a petri dish, and then we provide them with medium that is very similar to that of the nutrients that are moving through your bloodstream all the time. We then add those cells to our cultures of skin stem cells, and then we put them into this incubator. And this incubator then has the right environment, the right temperature, 98.6 degrees Fahrenheit, the temperature that our body temperature is in. And then, in addition, the right mixture of carbon dioxide and oxygen that allow us then, in this incubated environment, to be able to culture and maintain and propagate these cells. So this is what we do then. And if we now take a look at what happens after a week or so, after we've cultured these cells, if we grow these cells on plastic, and now we've stained these with hematoxylin and eosin, which allow us a histological stain so that we can visualize these stem cells, what we see is that these cells have the capacity to undergo division. And here you can see an anaphase cell, here you can see a metaphase cell, and here we can see other epidermal cells within the petri dish. And these cells divide then and can be propagated. We can passage them from one petri dish to another petri dish to another petri dish and keep getting more and more skin cells. In addition, we figured out ways in which we can recreate the environment so well that in fact the skin epidermis pretty much behaves and looks like a normal human skin epidermis. And in this case down here, what we've done is instead of plating these cells on plastic, instead we plate these cells now in a mixture of collagen and fibroblasts, which creates the dermis, and then we plate these epidermal cells down on that. We let them grow to confluence, and then at confluence we raise the collagen and plop it onto a raft, a floating raft, and now feed the cells from media below. Because your epidermis is at your skin surface, the blood vessels that are supplying the nutrients for your epidermis are actually located deep in the dermis, and those blood vessels then provide the nutrients, and that allows the epidermis to grow. And it turns out that one of the features of epidermal differentiation is basically predicated on the fact that the cells move away from their nutrient source. And so many years ago, one of my graduate students in the laboratory was studying some of the factors that are existing within the blood vessels and learned that in fact if you float these cells at the air-liquid interface, that now these cells can undergo a normal process of differentiation. And these cells up here would be the cells that in our body would be sloughed from the skin surface, continually being replaced by interlayer cells moving outward. But in a petri dish, these cells have nowhere to go. So these cells keep on piling up, and that's really about the only difference between this tissue and the skin epidermis of our body. So we can do pretty good about recreating the epidermis from scratch in a culture dish. So what can we use this for? Well, back when I was a postdoctoral fellow at MIT many years ago, my mentor, Howard Green, developed the technology for growing human epidermal cells in culture. And in fact, way back then, in the late 1970s, he and another investigator, Eugene Bell, also at MIT at the time, devised methods in which they could adapt the use of cultured human epidermal stem cells for burn therapy to treat badly burned patients. In many cases, patients' body surfaces would be so badly burned that there wasn't enough good skin to be able to graft. But if we took small amounts of the good skin and put those cells into culture, within a few weeks' time, we could generate many culture dishes of epidermal cells. And since they're the patient's own cells, the cells would not be rejected on a graft, and therefore we could take a small biopsy from the patient's skin, place those cells into culture, passage those cells, produce more large petri dishes full of skin stem cells, and then basically graft those cells onto the back, for instance, or the burned area of the victim. And then, since it's the patient's own cells, just passaged in culture, the patient doesn't reject them. They recognize the cells as self, and basically the burn is repaired. The cells that are coming from these petri dishes are epidermal stem cells. They are unipotent stem cells that generate epidermis. So the graft of these cells basically produces epidermis, but the patient's grafted skin does not contain hair follicles, nor does it contain sweat glands. But that's least of this patient's worry in a burn therapy. So the fact of the matter is that this type of technology then has been used across the world now in many various different burn clinics in conjunction with other classical means of skin grafting to be able to help patients and to be able to treat the most badly burned of patients. And I just point out to you that there are a number of biomedical engineering groups that are generating artificial skin. You might have heard of various different methods for artificial skin. You might have heard of various different doctors using pig skin or various different other types of non-human skin. Those skins are basically used as protective means until the patient's skin cells can be repaired, or for instance until these cultured cells can be able to take on the patient and regenerate epidermis. Pig skin cannot be used forever on a patient. That's recognized as foreign and would be rejected. Similarly, artificial skin is useful for a transient period of time, but basically one needs to have normal human cells in order to be able to recreate skin epithelium. So this is one of the few early on successful examples of the use of adult stem cells in a clinical setting. Okay, so epidermal cells can be grown for months in a dish and forever when grafted onto a burn patient. Just show you a few examples of this. So here is an example then that was taken again way back in the early days when people were just beginning to develop this technology for the use in regenerative medicine. And here's an example where the cells then were enzymatically removed from the petri dish once they were cultured, and then they were grafted on to this patient's badly burned leg. All of the cells that you see encompassing this entire area here were entirely generated from cultured epidermal cells from an area of good skin taken for this patient, placed into culture, and then grafted into the bad wounded area. Again, here's an example of one of the cultures. The cells that you see in pink here are the epidermal cells. And when those cells were grafted onto the patient's skin, here we see a cross-section of the patient's skin from the badly burned, what was a badly burned area, and now has perfectly healthy regenerated epidermis. And then if we follow this technology over time and say, well, a year later, how good is this epidermis? A year later, if you take a look at the epidermis of a normal patient and the epidermis of culture repaired epidermis grafted onto a person, a year later, you cannot tell the difference between the epidermis and the repaired epidermis from culture. The problem still is that there's problems with scarring. Problems with scarring are dermal problems. They're not epidermal problems. So scientists have long taken care of the epidermal problems using cultured epidermal stem cells for burn therapy. there's still a lot of work to go in the area of improving the quality of skin for burn patients, and in particular in the area of dermal repair and in the areas of generating hair follicles and generating sweat glands.